Hello, my name is Julian Yi. Today is February 23rd of 2023, and this is my presentation on the movement of macromolecules flowchart. So just to start off, I just want to do a little bit of background information. So we know that the four major macromolecules are carbohydrates, proteins, lipids, and nucleic acids, as we see on the bottom left picture. And we know that a macromolecule is a large molecule that is composed of different carbon-based molecules, and they typically compose all of life. So on the top right hand side, we see dehydration synthesis as well as hydrolysis. This is how we create polymers or break polymers down into monomers. So with dehydration synthesis, we see the formation of a polymer from two monomers by releasing H2O. And we add H2O to hydrolysis in order to break down two monomers. Now with the macromolecules, we see that carbohydrates are the primary source of energy, and they're typically linked by glycosidic linkages. We see proteins, and these are typically assisters with reactions and enzymes, and their bond types are peptide bonds. With lipids, we see that they are the secondary source of energy, and their bond types are ester linkages. And nucleic acid, which is our genetic info, they help convert genetic info into peptides, and they're typically phosphodiester bonds. So continuing on, we'll be looking at the steroid hormone. So for the steroid hormone, they are synthesized in the mitochondria as well as the smooth ER, and they start off as cholesterol. So here I have an image that I drew. So here is my cell. So the red is a cholesterol. So in the smooth ER, we'll have cholesterol, which will be released and put into a transport vesicle. And this transport vesicle will then move towards the Golgi apparatus, which is right here. So we get packaged, we'll get told where to go, and we will head towards the mitochondria. So here in the mitochondria, we'll be in the inner membrane. So in this inner membrane, we'll have a cleavage of a P450 enzyme, which will then convert cholesterol into pregnenolone, which would be this green right here. So this pregnenolone will be transported via transport vesicle and go back to the smooth ER, and this is where it will become a steroid hormone. And here, it will be released from the cell by a secretory vesicle by exocytosis. So when that occurs, we will then go to look at the release of progesterone. So here we have hypothalamus, which will release GnRH. After stimulation, it will release GnRH, go to the anterior pituitary gland, release FSH and LH, head towards the ovary and the uterus. So here we'll see that in the ovary, after FSH and LH develop, we will have the primary oocyte inside the ovary, and the theca and the granulosa cells will play a big part in how progesterone is released in the menstrual cycle. So how this occurs here, we'll be looking at this image in the back. So FSH and LH will be very stagnant, and once this burst comes, ovulation will occur and the corpus luteum will form. So when the corpus luteum forms, we'll have a very high increase in progesterone, which is we see right here. And with this increase in progesterone, we'll have the uterus become prepared for fertilization as well as implantation on the sides of the uterus. So as we continue on, LH and FSH levels decrease, and we see estrogen levels decreasing as well, and progesterone as the corpus luteum breaks apart. And when this occurs, the menstrual cycle will end. So now we'll be looking at protein, aka peptide hormones. So here I have an image that I drew, and we'll be seeing the ribosome right here, translating the mRNA into amino acids, which are later on proteins. And so here on the left-hand side, I have a cell, the nucleus, smooth ER, rough ER, vesicles, Golgi apparatus, and the membrane. So we go from the rough ER into the transport vesicle, which then go to the Golgi apparatus. And once we go to the Golgi apparatus, it will be synthesized and will be packaged. And here, it can make a decision. Do we want to stay in the cell or leave the cell? If we leave the cell, it will be a secretory vesicle, and it will be done by exocytosis. But here on the left-hand side, we can stay in the cell in a transport vesicle. And with this transport vesicle, it can wait for a signal or show some indication by the body that it needs to utilize this, and it will go about. So now we'll be looking at insulin. So with insulin, they start off in the pancreas, which is on the top right-hand side, and they are sitting in the beta cells of the pancreatic islets. So here we see insulin that is a triangle right up here. With this, it will connect to the insulin receptor on the cell membrane, and with this being done, it will allow for signal transduction to occur. And when this occurs, It will allow secondary messengers to release, which are CAMP, Ca2+, or nitric oxide, to help boost cell activation. And when this occurs, we allow glucose to come into the cell as well, and it will be utilized as energy, or it can be converted into glycogen in the liver, where it can be stored as energy when needed. So lastly, we'll be looking at intracellular proteins. So previously, we were looking at the peptide hormone slide. We will use the same cell as well as ribosomes. So we'll be translating the mRNA into amino acids, which is the building blocks of proteins, go in through the rough ER, or now we can utilize the free-floating ribosome, so we don't have to go through the ER or the Golgi apparatus. We'll be sit in transport vesicles already. Or we can go through the same process again, rough ER, transport vesicle, through the Golgi, and send a transport vesicle as well. So here we can utilize it for different things, such as DNA polymerase, the cytoskeleton of the cell with actin microtubules and intermediate filaments, or we can use it for the body through blood itself, which is because it's protein. So here we'll look at the blood circulation first. So here we see a hemoglobin that's made of 4 alpha or 2 beta chains, 2 alpha chains, and here we have in the circles oxygens connected. So here this will obviously travel through the whole body, through the circulatory system, and wherever needed it will help the respiratory system, or pretty much help everything in the body, allowing oxygen to flow. And imagine if we didn't have hemoglobin, then the body obviously would die away because no oxygen flow, no blood flow. So here, same thing with DNA polymerase. So here we have strands itself, we have helicase, DNA polymerase, and if we weren't to have these, then these would disrupt DNA synthesis, and we would probably be lacking some sort of DNA or some sort of chromosomal deficit. And here we see the leading strand, lagging strand, helicase, DNA polymerase to help split, and then DNA polymerase is formulating more strand, and SSBPs are helping keep them spread out and apart. So lastly, we will look at the cytoskeleton, and here we will allow more protein growth as well as more stability on the cell or even muscle filaments. So we have actin, microtubules, and intermediate filaments for those. The actin helps shape light shape the cell. The microtubules are major role in the cell reproduction cycle or the cell cycle, and the intermediate filaments help they're like the skeletal of the shape of the cell. So they'll help anchor in its position and prepare the cell wall to formulate. And that is all. Here are my works cited pages, and thank you.